Hello students, today we will see the significance of mitosis. In the previous video we have seen what is mitosis, how the process is undergoing in mitosis, how the cell undergoes from one stage to another stage. So the complete process of mitosis we have seen. What is mitosis? Mitosis is a type of cell division wherein the parental cell divides into two daughter cells carrying the same genetic material as the parental cell is carrying. So therefore it is known as equational division, and as it takes place in somatic cells in animals, it is also known as somatic cell division. So what is the importance of this mitosis or the significance of this mitosis? First, it produces diploid daughter cells with identical genetic complement. Yes, as the definition of mitosis says, it is an equational division and therefore it distributes equally its genetic material between its progeny cells, and also it distributes the same copy of genetic material without any variations as it was in the parent cell into the daughter cells. Mitosis cell division is required to maintain and restore nucleocytoplasmic ratio. What is this nucleocytoplasmic ratio? Because the cell growth is the property of cells, which includes increase in the size of the cell or say the number of the cells. If there is increase in the size of the cell, it is nothing but the nucleocytoplasmic ratio. That is, increases in the large cell or the cell undergoing growth is going to increase its volume of the nucleus and volume of the cytoplasm. So as in the cell that is growing, if it has to grow, it has to synthesize many metabolites. When it is synthesizing many metabolites, it grows in size in case of cytoplasm. So it becomes difficult for nucleus to control or regulate the cell. Therefore, to maintain this nucleocytoplasmic ratio, the cell undergoes division, which makes the ratio decrease, providing the nucleus all the comforts in ruling the cell. That is, maintaining the nucleocytoplasmic ratio. When the cell increases in size, the small size nucleus cannot control all the activities, and therefore it will undergo cell division, resulting into two daughter nuclei containing the cell contents. Growth of multicellular organisms is due to mitosis. Yes, that is, organism life start from a single cell that is known as zygote, which is formed from the fertilization of male gamete and female gamete. Now, this zygote, whatever is formed, is a single cell, and it is formed diploid cell, and it is formed from two haploid gametes, and therefore this zygote undergoes repeated mitotic cell division and leads to the formation of the whole body of an individual. So the whole body is developed from a single cell zygote. Mitosis cell division takes place in cell repair, that is, because of wounds there is a loss of cell, and because of any accident the cell is lost. All these cells are replaced by the mitotic cell division, because the same type of cells are produced by the mitotic cell division, because it is a equational division. Mitotic divisions will take place in meristematic tissues. In case of plants, there is a continuous growth through the meristematic cells, and therefore this continuous growth is brought about by the meristematic tissues that are present in the apical and the lateral cambium, as well as result in a continuous growth of plants throughout their life. In animals, the somatic cells undergo mitotic cell division to produce and to differentiate the cells and tissues and organs. So it undergoes repeated mitotic cell division in case of somatic cells in animals. In case of plants, these meristematic tissues allow plants to regrow structures or even form entirely new plants that results in vegetative propagation. So, to produce vegetative propagation, the cells undergo mitotic cell division. So, there are two types: apical meristems present at root and shoot tips responsible for primary growth, and lateral meristem occur at the cambium and are responsible for secondary growth. So these are some of the significances of somatic cell division. Now let us see the another type of cell division, that is meiosis. Meiosis is known as double division which occurs in a diploid cell. Yes, because it undergoes two types of division, therefore meiosis is known as double division. And how it is different from mitosis is, in mitosis, same replica of the chromosomes is transferred from one generation to another generation, whereas in meiosis the chromosome number is halved or reduced and it forms 4 haploid cells. In case of mitosis, 2 haploid cells are formed. In case of meiosis, 4 haploid cells are formed. Therefore, mitosis is known as equational division, meiosis is known as reductional division, because in the production of offspring by sexual reproduction includes the fusion of the two gametes, each with a complete haploid set of chromosomes. So these gametes are formed from specialized diploid cells through a process to form haploid cells. This process is known as meiosis. So meiosis is a specialized kind of cell division that is going to reduce the chromosome number of parent cell by half, resulting in the production of 4 haploid daughter cells. Right? So it is called as reduction division. And if you see where this meiosis occurs in diploid cells, generally at a particular time. This meiosis is not going to undergo, the cells doesn't undergo meiosis throughout their life. Only at certain period in their life they undergo meiotic cell division, and that too in specific diploid cells known as germ cells, or cells undergoing meiosis are called meiocytes. Cells undergoing meiosis are called meiocytes. Meiocytes are found in reproductive organs which are known as gonads and which are diploid germ cells. They have diploid number of chromosomes present. This diploid number of chromosomes is reduced to the haploid number of chromosomes in animals, as well as in case of plants also. So the germ cells in case of animals such as testis, which is a male reproductive organ, and this testis produces spermatocytes. This testis produces spermatocytes, and spermatocytes undergo meiosis, that is spermatogenesis, to form the sperms. And meiocytes found in female reproductive organs, that is in ovary, are oocytes. These oocytes once again undergo meiosis, and that is going to produce, the process of the meiosis of this oocytes is known as oogenesis, and it is going to produce a ovum or egg. Right? That is in case of animal cells. In considering the plant cells, in plants, in sporangia such as microsporocyte or microspore mother cells of anther, and megasporocytes or megaspore mother cells of ovule, of ovary, of flowers, form haploid spores. These are once again fertilized by the fertilization process and forms a single cell which is diploid in nature. So meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores this diploid phase. Clear? Now let us see what are some of the important key features of meiosis and how it is different from mitosis. Mitosis is single division and meiosis is double division, which involves two sequential cycles of division called meiosis I and meiosis II. In case of mitosis, interphase is single. Yes, it is same as in the case of meiosis also. It is going to remain single interphase stage where the replication of DNA is going to take place in the S phase. Right? And that too only once it is going to undergo replication, though there are two meiotic cell divisions in case of meiosis, but there is single interphase and single DNA replication in S phase is going to take place, and the diploid cell contains 2n chromosomes and amount of DNA after replication becomes 4C content. As usual, only the amount of DNA is going to double in the S phase, but the chromosome number is going to remain the same. Here, an extra additional step known as pairing of the chromosomes is going to take place, which is not taking place in case of mitosis. Okay, in mitosis, as such, the chromosomes, the chromatids present in the chromosome get distributed between the two daughter cells, whereas in case of homologous chromosomes, here they pair up and recombination of their segments is going to takes place. After the meiosis, true haploid cells are formed after both meiosis I and meiosis II process. After the first round of meiosis, two cells are formed with reduced chromosome number. These two cells further undergo meiosis II, which is similar to mitosis. It is going to produce four cells that are haploid in nature. As a result, four haploid cells are formed after the whole cycle of meiosis, that is after meiosis I and meiosis II. Four haploid cells are produced, which is different from its parent cell by half of its chromosome number. Let us see meiosis I. Now this meiosis I, we know that meiosis is divided into meiosis I and meiosis II. Meiosis I is further divided into karyokinesis I and cytokinesis I. It is divided into karyokinesis I and cytokinesis I. Karyokinesis is nuclear division, and this karyokinesis I is further divided into prophase I, metaphase-I, anaphase-I and telophase-I. And further, after karyokinesis or after nuclear division, once again the cell undergoes cytoplasm division known as cytokinesis I. Now the prophase I, it is not the simple prophase as that of the mitosis, but here the prophase is going to be more complicated and it is going to be a longest period of cell division. Right? The prophase I is the longest and more complicated stage compared to the prophase of the mitosis. Because this prophase is not a simple prophase, it is subdivided into 5 stages. Prophase I is subdivided into 5 substages such as leptotene, zygotene, pachytene, diplotene and diakinesis. Leptotene, zygotene, pachytene, diplotene and diakinesis. So the prophase I itself is further divided into 5 substages, which is not the case in case of mitosis. So what happens in this each of this stage? Let us see in detail. So what happens in leptotene? So once the cell after the interphase, in the S phase of the interphase, the DNA gets replicated, and in the G2 phase, whatever the biochemicals required, RNA are synthesized and other cell organelles are synthesized in the G2 phase. After this, the cell is going to enter into the leptotene of the prophase 1. So the DNA that is synthesized in the S phase of interphase is going to remain in the form of chromatin fibers. So in the interphase, it is going to remain in the form of chromatin fibers, and therefore in the leptotene stage, this chromatin fibers, they are going to condense to form elongated chromosomes. So the condensation of chromatin fibers is going to take place in the leptotene stage, that is thin thread like chromatin fiber structures are going to be thickened, shortened, elongated first because they are present in the coiled stage. They get encoiled, uncoiled, elongated, and then thickened and then shortened to form chromosomes. So the condensation is going to begin in the leptotene stage to form the elongated chromosomes. They were in chromatin fiber thread-like structures. Now they are transforming into the chromosomal structures. So that is what is going to take place in leptotene stage of prophase 1. Next, it is going to enter into the zygotene stage. Once again, one stage is not differentiated from the other stage by the clear-cut boundaries, but based upon the specific events that are going to take place in each event, one stage is being differentiated from the other stage. So after the chromosomes, the chromatin fiber is condensed into the chromosome form. Next, it is going to enter into the zygotene stage. So what happens in the zygotene stage? Here the condensation of the chromosomes, which was begin in the previous stage, that is in leptotene stage, is getting almost completed. The condensation of chromatin material to form chromosomes is almost completed in the zygotene stage. The cells DNA, now it is, which was in the form of chromatin fibers, is almost thickened, shortened, and it is in the form of thick chromosomal material. Now what happens? Here the step is going to be different as compared to the normal prophase of mitosis. In the normal prophase of mitosis, as the condensation is going to take place, in that prophase also the condensation is going to take place, but in case of prophase 1, an extra step is going to take place, that is pairing of homologous chromosomes. What are these homologous chromosomes? Now, in the S phase of the interphase, the DNA is replicated. Means each chromosome will be in the form of chromatin thread-like structure. So this chromatin thread-like structure is replicated. In S phase. Now, how many chromatin thread-like structures will be present? Now, each cell contains a diploid set of chromosome number, that is same pair. Same type of chromosome exists in a pair. Means single chromosome exists in a pair. That is, one set is derived from the paternal side and other set is derived from the maternal side. Now this paternal side set, that is, contains two chromatids, that is two replica of the chromosome known as chromatids, and from the maternal side it contains two chromatids. So together they are known as homologous chromosomes. Identical chromosomes are known as homologous chromosomes, that is derived from both the parents. So the pairing of these homologous chromosomes is going to take place. The pairing of these homologous chromosomes is going to take place, and that process is known as synapsis. That process is known as synapsis. That is, process of pairing of these homologous chromosomes is known as synapsis. You can see here in the diagram. These are the set of chromatids or a pair of homologous chromosome that is derived from the paternal side and here another pair of chromosomes that is derived from the maternal side. These are derived from the paternal side. So what happens, these were like thread like structures initially. They will be like this thread like structure initially in the S phase of the DNA. So as the condensation process has taken place, now they are forming a chromosome. This is known as a chromosome. Now each chromosome has a replica, yes or no? So these replica of this chromosome is known as sister chromatids. So one set is from parental side and they are both are replica of each other. They are known as sister chromatids. Another set is from maternal side and they are also having two replica known as sister chromatids. Right? Now these both are identical copies and therefore they together are known as homologous chromosomes. They are known as homologous chromosomes, and pairing of this paternal side and the maternal side chromosomes together is known as homologous chromosomes. So the process of pairing of the homologous chromosomes is known as synapsis. So during the synapsis, a structure called synaptonemal complex is formed. During the process of synapsis, a structure called synaptonemal complex is formed. This synaptonemal complex is made of nucleoproteins. This synaptonemal complex is made up of nucleoproteins and it contains a complex of proteins which contains a complex of proteins that are made up of nucleic acid and a protein material. So therefore it is known as synaptonemal complex, and the nucleoprotein complex helps in adherence of sister chromatids and then homologous chromosomes is called synaptonemal complex. It helps in adherence of sister chromatids together of the homologous chromosomes. So here if you see in the diagram, a synaptonemal complex is formed between these two homologous chromosomes, which is going to adhere these two homologous chromosomes together, and therefore the resulting structure or the homologous pair is called a bivalent or a tetrad. This resulting pair of the homologous chromosomes is known as a bivalent or it is known as a tetrad. Clear? So these are the things that is going to take place in Zygotene stage. So what all will take place? Condensation is almost completed, and then whatever the previously duplicated chromatin fibers in the S phase, they condense to form the chromatids in the sister chromatids in the Zygotene stage, and the pairing of this chromosomes, that is parental chromosomes, identical parental chromosome and the identical maternal chromosomes together, they are known as homologous chromosomes, and the pairing of these homologous chromosomes will result in the formation of a bivalent or a tetrad, and the process is known as synapsis. During the synapsis, a synaptonemal complex is obtained. A synaptonemal complex is formed, which is a nucleoproteinaceous complex that holds the two pairs of homologous chromosome together. Now the cell enters into the next stage, that is Pachytene stage. This stage is marked by the appearance of structures called recombination modules, and this Pachytene stage is a bit longer duration stage when compared with the Diplotene and Zygotene stage, and in this stage the tetrads are going to be visible much clearer when compared with the previous stages. So in the previous stage, these homologous chromosomes, they appear as bivalents, whereas in case of the Pachytene stage, they appear as tetrads in nature. Now this Pachytene is a longer duration, and you can observe this stage by the formation of certain structures known as recombination nodules. What are these recombination nodules in the tetrads of homologous chromosome? These recombination nodules are nothing but they are the sites near synaptonemal complex of tetrad, or these are the sites at which crossing over is going to take place. A process known as crossing over is going to take place between the homologous chromosomes. Now, this recombination nodules contain a multi-enzyme called as recombinase, which is going to participate in the crossing over process. Right. So, therefore, the recombination nodules are nothing but they are the sites where the crossing over is going to take place, and they are rich in multi-enzyme recombinase complex required for the process of crossing over. Crossing over is going to take place, it is a process that is going to take place between homologous chromosomes, and in the homologous chromosomes, that is between two non-sister chromatids. So the crossing over is a exchange of genetic material between two non-sister chromatids of a pair of homologous chromosomes by an enzyme recombinase. By an enzyme recombinase, it is known as crossing over. So what happens, see here, if you see this diagram, these two are sister chromatids and these two are sister chromatids. One is from paternal side and other one is from maternal side. Together they are forming homologous chromosomes. Okay, now the replicas of paternal side will become non-sister chromatids to the replicas of maternal side. Getting? These are replicas of paternal side. They are sister to each other, but they become non-sister to the replicas of maternal side. So the recombination is going to take place between the non-sister chromatids of homologous chromosomes. These two are known as non-sister chromatids, or this and this can be non-sister chromatids. This and this can be non-sister. This and this can be non-sister. This and this and this. So, different combinations can be obtained during the crossing over. So, the process of crossing over is known as exchange of genetic material between two non-sister chromatids of a pair of homologous chromosomes. It is mediated by an enzyme recombinase, and this recombinase enzyme contains two type of enzymes known as endonuclease and ligase. So the DNA in the between the non-sister chromatids is going to be denatured by the enzyme endonuclease, and some part of or some segment of the DNA is removed and attached to the another non-sister chromatid by the enzyme DNA ligase. So the chromatids of Pachytene chromosome are attached with centromere. In the Pachytene chromosome, they are attached with the centromere. So this ends the Pachytene stage. So once again, what are the events taking place in Pachytene stage? You can observe this stage as the formation of recombination nodules. Recombination nodules are the sites of crossing over where the crossing over has to take place, and they are rich in the recombinase enzyme, which is a complex containing an endonuclease enzyme and the DNA ligase enzyme, which is going to bring about the exchange of the genetic material between two non-sister chromatids of homologous chromosome. Clear? Next, the cell is going to enter into the Diplotene stage. Now all these stages are the substages of Prophase I. Still it didn't enter the next stage, that is Metaphase stage. So in this Diplotene stage, the homologous chromosomes or which were paired up in the Pachytene stage, okay, and which have already exchanged its genetic material in the Pachytene stage, now they begin to separate from one another. The pair of homologous chromosomes, they try to separate from one another, but they still remain intact at the sites of the crossovers. They still remain intact at the sites of the crossovers, and the sites of this previous crossovers which have already the crossing over has taken place, those sites resemble as X-shaped structures which are known as chiasmata. which is known as chiasmata. These chiasmata are the X-shaped structures and they resemble the sites of the previous crossovers. This stage, that is Diplotene stage, can be observed by the appearance of this chiasmata, whereas if you see the previous stage and synapses in the Zygotene stage, you can observe it by the formation of the synaptonemal complex, and in the Pachytene stage you can observe by the formation of recombination modules. Diplotene stage you can observe by the formation of the chiasmata, and it is seen only in the Diplotene stage. Now this terminalization of the synaptonemal complex is going to take place and the chromatids appear as tetrads. The terminalization of the means whatever the synaptonemal complex that has been formed in the Zygotene stage after the synapses, okay, which is going to held these two sister chromatids of parental side and the maternal side together. Now after the crossing over, slowly the synaptonemal complex is going to be dissolved and the chromatids appear as tetrads. Yes or no? They are going to appear as tetrads, and this stage remains for the long time, that is it remains for months or years together. The cells that are undergoing meiosis, they get arrested in this stage in majority of the cells for many years and months. For example, in case of the oocytes, in case of oocytes in the female reproductive organs produces oocytes, and these oocytes undergo oogenesis and through meiosis process, and this immature ovum or egg which is involved in reproduction are arrested in this stage for many months or years together. In the Diplotene stage, they reach this Diplotene stage in the 5th month of fetus and remain so for many years till the ovulation is going to occur, right? So the Diplotene stage can last for many months or years together. After the Diplotene stage, the cell is going to enter into the next stage known as Diakinesis stage. Now this is the last stage of the Prophase-I. Okay, so after this the cell is going to complete the Prophase-I and enter into the Metaphase-I. So the last phase of Prophase-I is known as diakinesis, and in this case the terminalization of chiasmata is going to take place. So in the previous step, that is in the Diplotene stage, the terminalization of synaptonemal complex is going to take place, but here the terminalization of chiasmata is going to take place. What is this terminalization of chiasmata? It is movement of chiasmata from their original position and pass outwardly. Means whatever the chiasmata is being formed between these homologous chromosomes, that is that too between the non-sister chromatids during the after the crossover has taken place, they slowly move from these chromatids and move outside of the cell, and that process is known as terminalization. By the end of this stage, chromosomes are fully condensed. The condensation was continuing in all the stages, began from the leptotene stage and is going to complete in the diakinesis stage, and now the chromosomes are fully condensed and they are clear and they can be seen in the metaphase stage. Here, as it is in the prophase of mitosis, the nuclear envelope and nucleolus is going to disappear, and the formation of meiotic spindle fibers for the separation of the chromosomes in the metaphase stage are initiated at the centrioles in case of animal cells and from the microtubules in case of plant cells. So the formation of meiotic spindle fibers begin for the separation of these chromosomes. So these are the events that is going to take place in the diakinesis stage, right? That is, it is the last phase and the terminalization of chiasmata has taken place, and by the end of this stage chromosomes are fully condensed, nucleolus and nuclear envelope is going to disappear, and the formation of meiotic spindle fibers begins for the separation of homologous chromosomes. Now the cell is going to enter into the Metaphase-I stage. So what happens in Metaphase-I stage? The spindle fibers initiated from the opposite poles attached to the kinetochores of pair of recombined homologous chromosomes. Spindle fibers that are initiated from the opposite poles, they are going to, it is similar to that of the metaphase of the mitosis, they are going to attach to the kinetochores of the chromosomes in case of mitosis. Here kinetochores of the pair of homologous chromosomes, because the homologous chromosomes were bit attached at the site through, though the chiasmata is terminalized in the diakinesis stage, but also at some sites they are still attached, right? And then at the spindle fibers contract, these chromosomes are moved over the equator of the spindle apparatus. They are moved towards the equator of the, means at the equatorial region, these spindle fibers are going to bring the chromosomes. Homologous chromosomes are brought to the equatorial region, and that process of bringing these chromosomes to the equatorial region is known as congregation, and the arrangement of these chromosomes at the equatorial region will form double metaphasic plate. In case of mitosis, in metaphase it is going to form an equatorial plate, or it is known as metaphase plate. Now what is this equatorial plate or metaphase plate? When the chromosomes are aligned at the equatorial region, it is known as equatorial plate or metaphasic plate. Here in the metaphase 1, there is no single chromosome, but a pair of chromosomes are aligned at the equatorial region. Therefore, it results in double metaphasic plate is going to be formed, right? And in anaphase 1, here now the chiasmata, though a slight chiasmata was found in a metaphase stage, in case of anaphase the complete disappearance of chiasmata is going to take place, leading to the separation of homologous chromosomes known as disjunction. It is known as disjunction. So the homologous chromosomes are pulled by these spindle fibers. Whereas in case of normal anaphase of metaphase, the chromatids of the chromosome are pulled to the opposite poles, means the centromere holding these chromatids together is split and taken to the opposite poles. But here a pair of homologous chromosomes are getting segregated, and each pair is separated and pulled towards the opposite poles, and the homologous chromosomes are separated, but why the sister chromatids are still held by the centromere. There is no splitting of the centromere taking place in anaphase 1, but only the homologous chromosomes are separated from their chiasmata and they are pulled to the opposite poles. So now the opposite poles contain number of pairs of homologous chromosomes. Either it could be recombined paternal or maternal chromosomes. So once again they follow the law of Mendel's independent assortment, and each homologous chromosome with their recombined part, because which have been undergone recombination in the Pachytene stage. Now the paternal chromosome contains some of the part of the maternal chromosome, and the maternal chromosome contains a recombined part of the paternal chromosome, and they get segregated independently and move to the opposite poles. So here the chromosome number is reduced to half, resulting in haploid cell, because here the chromosome is not splitting its chromatids, and chromatids are not moving to the opposite pole, but the whole chromosome itself is moved to the opposite pole, and therefore it results in reduce in the chromosome number and results in the haploid cell. And the last stage is telophase 1. So in the telophase 1, it is an optional stage in meiosis 1. So because after anaphase 1, the cell undergoes telophase and cytokinesis simultaneously, but sometimes it undergoes separately. So it is known as an optional stage in meiosis I. Once again, it is similar to the prophase stage of telophase of the mitosis. Like once again, the nucleolus and nuclear envelope is going to reappear, and it is followed by cytokinesis, leading to the formation of dyad of cells. Means it is going to contain, each chromosome is containing once again two chromatids at the opposite poles, and the chromosomes decondense but not exactly as same as interphase nucleus. Means once again the chromosomes decondense back to form the network of chromatin like structures, but in case of telophase I, it is not that complete. That in case of interphase and in case of prophase, how the cell is going to have chromatin, it is not going to be decondensed to that extent, but they are still in the elongated form only, right? So after telophase I, the cell enters cytokinesis I. Once again, it is going to produce or undergo cell furrow method or cell plate method, and divides the cytoplasm and forms two daughter cells which are haploid in nature. It is going to form two haploid cells, that is two cells that is going to be haploid in nature.